Let's talk about what happens when mitosis goes awry. So there's something called the cell cycle control system, and this is a cycling set of there's a lot of growth, there's something called growth factors which help signal the cell cycle um, to go forward or to stop, to regulate the cell cycle. Um, so the cell cycle control system is a cycling set of molecules in the cell that triggers and coordinates key events in the cell cycle. That includes the phases of interphase and the phases of mitosis. Checkpoints in the cell cycle can either stop an event or signal an event to proceed. And by events, we're specifically talking about stages or things that happen in mitosis and interphase. So checkpoints are really important because you don't want your cells to just constantly divide. You just want them to divide when you need new cells. I mean, your cells are dividing a lot when you're growing, but then you reach an adult height and you don't want to keep growing. You want to stay your normal adult height so you don't have health problems. Certain areas of your body don't, like once your fingers are formed, you don't want to keep them to keep getting longer, right? You want to have the right amount of blood, not too much, not too little. You want to have the right proportions of body organs and anatomical features. And then, so once you're an adult, you only want your cells to divide when your cells need to be replaced, right? So all your cells have these, they're not always going through mitosis. They only undergo cell division to make new cells when they need to. And the checkpoints are points in the cell cycle where the cell checks on its surrounding environment and decides whether it needs to, make new cells or not. The cell cycle checkpoints are cellular mechanisms. They regulate the cell's progression through the cell cycle, and they ensure each stage is completed accurately also. So not only is the cell checking to see whether it needs to make daughter cells, whether it needs to divide, but it's also making sure, it's also self-checking, proofreading DNA, making sure that everything up to that point has happened correctly. All right. So checkpoints prevent a cell from progressing to the next stage until it accurately finishes the current stage it's in. So there are three major checkpoints in the cell cycle. There's the G1 checkpoint, which allows entry into the S phase, or causes the cell to leave the cycle completely, entering a non-dividing G0, G0 phase. Right. So there's a G1 checkpoint which says, do I need to pause here in G0 or do I continue on to the S phase? There's a G2 checkpoint that says, do I need to pause here or do I need to continue into the mitotic phase? And then there's also a checkpoint during mitosis. So research on the control of the cell cycle is one of the hottest areas in biology today, and you'll see why in just a moment. So all the cells within an organism's body divide and develop at different rates. Some cell types need to be replaced regularly, and some don't ever need to be replaced. So cell division is controlled by environmental signals. There's three main environmental signals. There is anchorage dependence, density-dependent inhibition, and growth factors. So anchorage dependence is the need for cells to be in contact with a solid surface to divide. So if like a cell on the surface of an organ gets pulled off, dislodges, and is floating in the bloodstream, it should not divide because, that would clot your capillaries or maybe even larger vessels if it continued to divide while it's floating around. So for cells to divide, they need to be attached to a surface, a solid surface. So that's anchorage dependence. Density-dependent inhibition. Cells can sense how crowded they are. So they can sense whether they're getting pushed up against other cells too much or not. And when they're really crowded, they should stop dividing. They need the presence of essential nutrients to divide. So if nutrition is not optimal, then that will interfere with mitosis happening appropriately. But also growth factors, which are proteins that stimulate division. And they're proteins that are sent out, they're communication molecules sent out from other cells that might say, they might be certain types of hormones, for example. So your hormones can stimulate growth, right? So so growth factors are hormones or other kinds of proteins that stimulate mitosis at the right time. So they're sent from one part of the body to another. So here's an example of the effect of growth factors. So here you have cultured cells that are suspended in liquid. You put them onto a solid surface. These cells fail to divide. They need to be in the presence of a growth factor in order for them to divide and make new cells. So here's the cell cycle down here. You start in G1, go through S phase, go through G2, and go through mitosis. And these traffic signals represent where you find the 3 different checkpoints on the cell cycle. So the cell stops here, looks for the correct signals before it proceeds into S phase. Then it stops here, looks for the correct signals before it enters into mitosis. And then it can even be paused in the middle of mitosis before moving on back into, before completing division and moving into G1 again. And so this diagram shows the importance of growth factors. So growth factors are sent by other cells, like there might be some kind of hormone that signals, it's indicating the cell that growth, it's time to grow. It's time to make new cells so this organism can grow taller, can grow bigger. So this harkens back to the signal transduction pathway. So you have a ligand, which in this case is a growth factor that lodges in the receptor protein. Receptor protein changes shape, goes through some kind of conformational change, and then becomes relay proteins. It goes through this whole signal transduction pathway, and the end result in this case is to go past this G1 checkpoint. So once at the end of the signal transduction pathway, there's a signal for the cell to continue through the cell cycle and replicate its DNA. Go past the S phase and G2 phase. All right, so that's how cells communicate with each other. That's how a cell can figure out when it's time to divide or when it's time to stop dividing. So what about so now let's talk about mutations. What happens when this goes wrong? So a mutation is an incorrect DNA code. It's caused by an error made by DNA polymerase during DNA replication, right? So DNA polymerase has some proofreading capacity, but it doesn't always catch its own mistakes, especially if the DNA is damaged, so it's hard to read. It's hard to figure out exactly what the new copy is supposed to say. So carcinogens and mutagens in the environment, like UV light and certain types of chemicals, they can damage the DNA and make it hard for DNA polymerase to read the existing strand correctly when it's building a new strand. But also sometimes DNA polymerase just makes mistakes. So when DNA polymerase does not build the new strand correctly, when it puts the wrong nucleic acid in, when it replaces a T with an A, for example, that's a mutation. And this, of course, happens during S phase, because S phase is when DNA is replicated. So mutations happen during S phase while the DNA is being replicated. This can result in faulty proteins because, as you learned about with transcription translation, you did that activity where you could see the effect of different types of mutations on the resulting proteins. So mutation in DNA code can result in faulty proteins. So what if a cell experiences a mutation in a region of DNA that codes for proteins involved with checkpoints? What happens then? Well, cell division is really tightly regulated. When a mutation to a gene involved with checkpoints happens, usually the cell can either repair that damage at the checkpoints. So remember, the cell cycle checkpoints involve a self-check often. So the cell can proofread its DNA yet again and repair the damage, or if too much damage has happened and it's irreparable, the cell can undergo apoptosis or commit suicide. This is a sacrifice the cell makes to the health of the whole organism. Because if you have a cell with damaged checkpoints, that means that its cell cycle isn't regulated and might divide too much, and that can really compromise the health of the organism. So rather than putting the whole body at risk, this one cell will kill itself, will undergo programmed cell death, which is called apoptosis. When a normal cell sustains irreparable damage, it undergoes programmed cell death, and this cellular suicide prevents cells from producing more damaged daughter cells. But if there's enough DNA damage to interfere with multiple checkpoints, if the cell doesn't catch it with proofreading, or the mutation happens so that the cell can't proofread, because there are proteins involved at the checkpoints that are responsible for proofreading. So it's really bad if you get a mutation for a proofreading protein, because then the cell can't proofread for errors. And that means that these errors get by, and the cell doesn't repair them or doesn't undergo apoptosis. So if there's enough DNA damage to interfere with multiple checkpoints, the result is cancer. because cancer is when cells divide uncontrollably and a tumor forms. So a tumor is just a tight cluster of cells that are dividing uncontrollably, growing bigger and bigger and bigger. So a mutation in a checkpoint can prevent a cell from identifying and repairing other mutations, which means that there will be a faster accumulation of mutations after that first mutation. Right? Because the cell is repairing mutations all the time, but if you stop its proofreading capabilities or stop its repairing capabilities, then those mutations won't be repaired, and so you'll get more and more mutations accumulating if you disrupt the proofreading capabilities of the cell. So there's two different types of genes involved with cancers. Proto-oncogenes are genes that normally promote cell division and differentiation. They can be mutated to become permanently activated. So proto-oncogenes are supposed to be turned off some of the time. And then when it's time to divide, the proto-oncogene gets turned on. But if they are mutated so that they're on all the time, so that the light is always green, that's an oncogene. So you want all of these signals to be sometimes red and sometimes green, right? So proto-oncogenes give the go-ahead for cell division. They stimulate cell division. They should be off when it's not time to divide, but when they're turned on, if they're turned on permanently, then the cell divides continuously. And when they're normal, they're proto-oncogenes. When they're permanently turned on because of a mutation, they're called oncogenes. Tumor suppressor genes normally pause cell division in order to repair DNA, or they can initiate cell death. So they pause cell division, they repair DNA, and or initiate cell death. Once mutated, they can become mutated so that they're always inactivated. So that if these genes never turn on, then that means that the cell is never pausing its division, it's never repairing its DNA, or it's never initiating cell death. So some breast cancer genes are tumor suppressor genes that have been mutated. So BRCA1 and BRCA2, these are genes that are implicated in high risks of breast cancer, and these are tumor suppressor genes that are mutated so that they're not working properly. So these are the kinds of mutations that prevent a cell from self-checking and repairing mutations so that further mutations are more likely to occur. So here's an example of what would happen if some of these kinds of genes, so for example, tumor suppressor genes or proto-oncogenes some of them can help sense when the cells are too crowded. So here's normal cells. They're not crowded here and they're anchored. So once they're anchored, they should divide until they form a single layer of cells, and then they should stop dividing. When you remove cells, that's like creating a wound. They'll divide again to fill in the space, and then they'll stop dividing. Genes where proto-oncogenes or tumor suppressor genes are damaged, they're not able to stop cell division once the cells are crowded. So they're not processing the crowding signals correctly. So these cells are receiving signals from their neighbors. They're receiving signals that they're crowded, and that stops cell division. Here, the signals of crowding are not being processed correctly to stop cell division, so the cells just keep dividing. These are cancer cells. So that's what a tumor is. So that's the kind of thing that happens when tumor suppressor genes and proto-oncogenes are mutated so that they don't work properly. So if proto-oncogenes are permanently turned on, they're always dividing. Tumor suppressor genes are supposed to stop cell division. For example, when cells are crowded, it's supposed to stop cell division. If the tumor suppressor gene is damaged, it won't be able to stop cell division. All right, so cancer is a disease of unregulated cell division. Cells divide inappropriately and accumulate, in some instances forming a tumor. Treatment can be surgery if the cancer is contained to one location in the body. But if a piece of the tumor breaks off and travels in the bloodstream to other parts of the body, that's when the cancer is metastasized, then you can't use surgery anymore to treat the cancer. So that's when you use chemotherapy. So cancers kill by crowding out normal cells, invading other organs, and secreting poisonous chemicals. So sometimes, remember I said that if a tumor suppressor gene is damaged, the cell can't repair itself from further damage. So sometimes mutations happen that cause the cancer cell to produce toxins and things like that because of mutations that are no longer being caught by the proteins coded by tumor suppressor genes. So sometimes cancers secrete poisons because of those mutations that are collecting. So cancer cells start out as normal body cells. They undergo genetic mutations that cause them to lose the ability to control the tempo of their own division, and so they cause disease. So cancer therapy seeks to disrupt one or more steps in cell division. So chemotherapies disrupt mitosis, and that's why they're so toxic, because all of the cells in your body need to use mitosis at some point. You need to replace old and damaged cells. So if you're disrupting mitosis, you're disrupting the spread of the cancer, the growth of the tumor, but you're also disrupting the normal division processes of all the cells in your body. So that's why chemotherapy is so toxic to a patient. It's not a very elegant method of dealing with cancer. We hope to get better ones. It's really playing a game of chicken. All right, you're going to stop a person's cells from dividing and hopefully kill the cancer cell before you kill the patient. So it's not very elegant, but it's all we have right now for managing cancer.